And we're going to start with, okay, we're going to start with Gustavo, and then Itziar will go after him. And just a reminder, so if you have any questions about TICA, you want to be a member, you want to invite someone to be a member, our website is composite.org. If someone can type that on the, I'm going to type that on the chat. And we also have a YouTube channel where all the talks are posted after like probably a week or so after each talk. And all the videos from the Composite Morphology workshop that happened during the Botany 2021 event this year are also posted on this YouTube channel. They are like a amazing resource for a lot of people. So if you guys want to watch it and share, feel free to do that. I'm going to pass the link to the YouTube channel in the chat too, but you should have received it in the email with the Zoom link for this meeting. Okay, so to start with, we're going to have Gustavo Hayden. He's a researcher at Embrapa, which is the Brazilian agronomical research enterprise, basically. And he has studied Baccharis since he was an undergrad, and he's done like his life work in studying this mega genus. He went to study that on his master's, on his PhD, and now at Embrapa he keeps on studying Baccharis, but he also works with genomic resources of crop relatives in Brazil, which is a very interesting work in itself. And I'll let him start. I hope you enjoy this talk as much as I enjoy his other past talks.
Hello, Carol. Hello, everyone. Are you listening well? Okay. So in first place, thank you very much for the invitation, especially for you, Mauricio, Jennifer, and Isaac for organizing all this cool stuff that's happening in TICA Talks. It's a pleasure to be here today. And then Carol already introduced myself. So I am a botanist from Brazil, working on the Brazilian Agricultural Enterprise Corporation. That's more or less like the USDA, for example. And I'm based in south of Brazil here, in the south of Brazil, close to the Uruguayan border. I'm a neighbor of Mauricio. So here is a view of my research station and the herbarium where I am based on. So I'm going to talk about Baccharis. It's a panorama of Baccharis, the genus that I'm studying since a long time, more than 15 years ago. I started researching and investigating this genus. I had a lot of curiosity about some plants that had winged stems. I started with that plants, and with the passing of time, I expanded my scope. And currently, I'm dealing with the whole genus, mostly on taxonomy and systematics, but also working in collaboration with other people from several different areas, because it's always fun to get out of our comfort zone and try to find new horizons, to find new applications for the plants we study. So here in this previous slide, I forgot to tell you, this is Baccharis ciliata, a very narrow endemic from the Organ Mountains in Rio de Janeiro state. It occupies an area that's less than 100 square meters. It's very restricted. People, when think about Baccharis, generally think about some widespread species that are very common, but almost half of the species of the genus are narrow endemics or endemics. And this is something that I always try to highlight because it has a lot of meaning for conservation of the diversity, plant diversity. So, as you know, there are several megagenera in Asteraceae. Some of them are not so mega today because they were split in smaller genera, depending on the point of view of the systematics. And this makes Baccharis one of the top 10, or depending on the circumscription of the other genera, one of the top 5 most specialized genus in the Asteraceae. Just like Micania, Baccharis has around 450 species. What I'm going to tell you today about Baccharis is mostly based on three publications. One article from 2019 about the phylogeny of the genus, another article in collaboration with Mauricio, telling about the overall aspects of Baccharis and the most striking features of the genus, and another book chapter that has an updated checklist with all associated distribution data for countries, territories, and also for subdivisions of some large countries. So this data I'm going to present, it's based in this updated checklist that of accepted species, that all the species are classified into sections or subgenera based on molecular or morphological data. This work is also the results of more than the review of several herbaria with more than 100,000 specimens studied till today, and a lot of field work too from several areas that are depicted in this map here on the left, where we can see sampling sites from the northeastern USA to almost the border, the beginning of Patagonia. Plus the sampling did by myself. I also have something for the phylogeneticists that were provided by other collectors too. And so far I have collected more than 1,300 specimens of the genus. And I have now about 700 samples for that. Some of them were already extracted and sequenced, and so others were not because they are like duplicates of same species or are waiting to be sequenced. So far, since I started working with the genus, 70 new species were described. DNA extraction was done for almost 400 samples. And I'm working based on the phylogeny is based mostly on the sequencing of 4 regions of DNA, 2 from the nuclear genome and 2 from the chloroplastidial genome. In the phylogeny published in the paper I mentioned in the beginning of the presentation, 248 species were included in that phylogeny with the 4 markers sequenced, and 44 outgroups from the tribe Aster were also analyzed together in that phylogeny by the mean of Bayesian phylogenetic inference. In this slide, I depict some of the new species that have been described so far along the course of this work. So this is how the overall phylogeny looks like. So based on this work, we could confirm that 5 generic segregates could be synonymized within Baccharis to make Baccharis monophyletic, and 2 genera that were then synonymized, they are now recognized as subgenera, and 2 sections of genus Baccharis were now considered as subgenus. They were turned into a higher taxonomic status. The previous classifications had circumscriptions for 3 subgenera, Baccharis, Molina, and Tarchonantoides, that were not monophyletic. So after the phylogenetic analysis, we could recircumscribe this subgenera to reflect the evolution. And 2 subgenera were synonymized to Baccharis subgenus Molina. So the current infragenetic classification recognizes 7 subgenera within Baccharis. This beautiful slide are the result of the work, the design work by Mauricio. So Mauricio turned that previous simplified phylogeny based mostly on the subgenera into a sectional level diagram showing the relationships among sections within each subgenus. So each subgenus has a distinct color in this image, and we can see so that we have two big stars that one is represented by several sections of subgenus Molina and some sections of subgenus Baccharis, and plus some species of these both subgenera that were not sampled for molecular sequencing and were not included in the phylogenetic analysis. But these 2 groups of species, they could be placed in each following subgenera based mostly on morphology. After this work was finished, we could then test if some morphological traits commonly used to circumscribe the genus, they were synapomorphs or not for the whole genus. So we confirmed that the homogamous capitulum with staminate flowers and pistillate flowers, generally in distinct individuals, could be considered as the most important synapomorphy of the genus, because it's shared for most of the representatives, with some special cases of reversions or variations on the dioiche. And we could also show that the tufted indumentum, that's a type of trichome arrangement exclusive from Baccharis, and that it was considered a synapomorphy for the genus. It's not a synapomorphy for the whole genus, because this special tufted indumentum, it appeared just after the split between subgenus Tarchonodontoides that lacks this type of indumentum and the remaining subgenera. This type of indumentum, that is, it can be seen as minute dots hidden by the other trichomes on the surface of leaves of Baccharis, like this minute dots here. They are in fact several trichomes that are fused into a single basal cell. And this type of indumentum then is absent in the first diverging lineage, and it also is absent in more derived subgenera, or even in some scattered species along the phylogeny. So it's not a synapomorphy of Baccharis anymore. Dioiche was also considered a synapomorphy of the genus, but then with the phylogeny, we could see that other types of sexual expression of Baccharis, of genera that were segregated from Baccharis as Eperotalamus, Baccharidastrum, and Baccharidiopsis, they were in fact deviations of the main pattern of dioiche. And then what we consider that's the synapomorphy of the genus is the florets that are functionally only staminate or only pistillate. Only one species of the whole genus has a report of hermaphrodite flowers, but these should be further investigated. All the remaining species, they have unisexual florets that could be distributed in different specimens or in different arrays depending on each group of species. So we could have polygamous species, monoecious species, gynodioecious species, and most of them are dioecious. This plate, also prepared by Mauricio, depicts several important characters to identify the species of the genus, or even to get to higher groups of species like sections or subgenera. So when we are working on Baccharis, it's important to evaluate the type of capitulum, if it's homogamous, if it's heterogamous, if it's disciform, if it has ray florets, the shape of the lobes of the staminate florets, and the length of the tube. It's also important. For the pistillate florets, we have a huge variation with short rayed corollas, dentate, truncate ones. We have some truncate ones that have like a red or trichomes just a bit below the apex of the corolla. And sometimes it seems like they are lobed, but it's not, it's trichomes in this case. We also have a lot of variation on the styopips of mainly male florets, that they could have different outline shapes when we are looking for subgenera. For example, some subgenera has this type that's more linear with free branches. Some have these branches that are linear but fused. Some have like rhombic apexes. And other useful features are the pappus. There are some species that have a lot of variation when you compare the female, the pistillate plant pappus, with the staminate plant pappus. So the staminate florets, they usually have very broadened apical ends. Meanwhile, the pistillate florets, they have mostly scabbard pappus elements. It's also very important to, when we are identifying mainly sections, the number of ribs around the cypsella. So there are species with less than 5 ribs, There are other with between 5 and 8 ribs mostly, and there are other groups with more than 8 ribs. When you do a cross section, it's easier to count this number of ribs, for example. And this other plate, it also depicts how these different arrangements of capitula are distributed and represented within the genus. So the most common type with perfectly dioecious species, we called it a Halimifolia type, and it's more than 400 species that presents this pattern of distribution with capitula with only staminate and the other with only pistillate and only staminate florets, for example. And then we have the other types of arrangements that could be very, very distinct from this main pattern. So this is how these different types of arrangements of florets looks like in fresh material. Here in A, we have only pistillate corollas. Here only staminate florets with laciniate apex. Here we have a capitulum that has a central staminate florets and marginal pistillate florets. Here it's in D, it's a capitulum with only short-rayed pistillate florets. And here it's another capitulum from the same species that has short-rayed ray florets and also tubulose disc florets. So the genus has a very wide distribution across the whole continent of the Americas, and it occupies a large array of habitats. This reflects in the morphology. Most of the species are shrubs, but we have some herbs, subshrubs, trees, and even climbing species. And the habitats occupied varies a lot from grasslands to coastal dunes, lake shores, savannas, the yungas in Bolivia, deserts, semi-deserts, and the Altandina vegetation are some examples of habitats occupied by Baccharis. So we can have species growing by the sea level or even reaching more than 4,500 meters above sea level. This is the distribution of Baccharis across the American continent. So we have Baccharis halimifolia in its northeasternmost point in the Nova Scotia Island in Canada, Baccharis pilularis on the northwestern coast of USA, Baccharis magellamica on the southernmost islands of South America, and here in the easternmost point of natural distribution of Baccharis, we have Baccharis cinerea on the coastal scrubs of eastern Brazil. We also have some species that were introduced on purpose or by chance in other countries, and two of them are of special interest. Baccharis halimifolia became an invasive species in several Mediterranean and the North Sea coast of Europe. It caused a lot of damage of habitat disruptions in coastal environments there. The same happened here in Australia and New Zealand. And recently, Baccharis spicata was also introduced in Portugal. And the monitoring of this population showed that they are stable and they are now trying to eradicate it before it becomes a problem as high as halimifolia is today. Well, so this the distribution of Baccharis is unevenly across the whole continent. We can see here that most of the species are found in southeastern Brazil. It's the main center of phylogenetic and also species diversity. But the Andes from Bolivia to and Peru mainly, but also including Colombia and Ecuador and northern Argentina and Chile, are also very important for species richness of Baccharis. Brazil has currently 185 species recorded, and more than 100 of them are endemic, followed by Argentina, Bolivia, Peru, and Uruguay. I highlight here because it's a very small country compared to the others before, and it also has more than 10% of the genus diversity. The United States is also remarkable. It has 23 species and 4 of them are endemics. And Ecuador, that we're gonna need to know a bit better after my talk with Itziar, also has a very high diversity with 38 species, and 10 of them are endemic. So in this image, I show the 4 most widespread species, Baccharis dioica, that occurs in several islands along the Caribbean and also in the coasts of Mexico and United States. Baccharis pedunculata, found in Mesoamerica, south to northern South America. Baccharis salicifolia, that occurs from the United States until the southern tip of Patagonia. Baccharis trinervis, that's widespread in several forests in South America. But as I said before, most of the Baccharis species, they are distributed in much narrower areas. And here I bring some examples from every different subgenus, like Baccharis chionolenoides, Baccharis friburgensis, Baccharis magnifica, Baccharis cinerea, all of them occurring in very restricted areas. Here we also have Baccharis wagenitzi, Baccharis pluricapitulata, Baccharis scopulorum, and Baccharis obidotata. Most of them are only known to places very restricted, like just a single mountain summit, for example. So all this has been done till now concerning the knowledge of the genus, but what's next? What's still to be done? And there is a lot of work still to be done within concerning Baccharis. I'm focusing now on revising the taxonomy of the smaller subgenera that were already fully or almost fully sampled in the phylogeny. So the taxonomy review of Baccharis subgenus Tarconantoides is finished. For the subgenus Oblongifolia, I would say that's halfway done. For subgenus Eperotalamus, I could get new samples from some species that were missing in the first phylogeny, and I hope to get more resolution for this group and start dedicating more efforts on understanding its evolution. This is an example of the already published taxonomic revision of Baccharis subgenus Tarconantoides. So it was possible to identify synapomorphisms for each clade based on morphology. Here is the distribution of this subgenus in East, Southeast, and South America. And it's a classical taxonomic work that we have field work pictures, habitat pictures, line drawings to aid identification, distributional maps, preliminary conservation status, folk names, phenology. And the idea is to publish similar studies for the remaining subgenus. This is an example of some species of subgenus Oblongifolia. The review of the Brazilian species is already finished, but in northern Andean countries, there is a lot of work still to be done to understand the species boundaries there. This is a plate depicting 2 species of this subgenus. Here, I'm showing the other 2 subgenera that are under taxonomic studies. One is the subgenus Eperotalumalopsis that was previously considered a segregated monospecific genus, and now it's placed here in the phylogeny. So far, it only has 1 species known. The other is the subgenus Coridifolia that has 10 species in 2 sections recognized. So this is how the manuscript of Coridifolia looks like. I would say I still have to get plates for half of the species, but the new species of this section are already published and the descriptions are finished. We have a huge sampling gap in the subgenus Molina. This subgenus is very variable. We can see here herbs, shrubs, geophytes, climbing species, and the resolution of the phylogeny in this subgenus is not so good. So some values are not very good, are not good. And mainly, I believe that a lot of sampling is lacking here. This is the subgenus last sampled, and mainly because of it has a lot of Mexican endemic species, and I don't have so far any sample of Mexican species in the phylogeny, for example. The other subgenus that miss a lot of something yet is subgenus Baccharis that accounts for half of the species. and it has this current classification in sections. The type species of the genus, Baccharis alimifolia, here seen growing in the Central Park in New York. It's the type species of the genus. It has 17 sections, only 13 of them were already sampled for the phylogeny. And one interesting result from the phylogeny was that the group of species with alate, with winged stems, it was recovered as two different clades, and the evolution of these groups is very intriguing. In the section Colopteri, all the species, they have winged stems and most of them have reduced leaves. But when you go to section Aphili, we have wingless species, leafless species. We have species that have developed leaves but no winged stems. We have species that have winged stems and developed leaves. And the variation of other traits is also very high within this section, but we also have characteristics in the cypsela of each section that give support to this distinction into sections. Another groups that are well sampled are the sections Agglomerati, Axillaris, and the last one is Cuneiforli. I typed it wrongly here. It's Cuneiforli down here. And these sections, they have some new species that I'm describing with Caetano Oliveira from Minas Gerais that are very remarkable and are still to be sampled for the phylogeny. The analysis on subgenus Baccharis also allowed to recognize some groups, some sections not previously recognized before, like section Irinite with only 2 species from Paraguay and central Brazil, and section Andina from the northern Andes, and with a lot of species found in Colombia, Ecuador, Peru mostly, and also Venezuela. So the data obtained till today, we can say that 442 species are recognized in Baccharis. They are classified into 47 sections and 7 subgenera. But so far, 12 sections and almost 200 species were not sequenced yet. So one of my goals is to increase the sampling of these missing terminals for the phylogeny. I already got more, I would say 60 species beyond that this first phylogeny. But my goal is to, when reach 100 new terminals, to publish a novel, a newer phylogenetic classification, moving from 55% of sampling to 75% of coverage. As it's written in this Bolivian coin, the union is our power. So all this work was not done alone. It's all the result of collaboration with several people. Several of them are here attending this presentation today. And for example, a lot of the newly described species were done in collaboration. This is Baccharis funky, recently described by Mauricio, myself, Victoria, and Eduardo Marchesi from Uruguay. And we honored Vicky Funky, so with this beautiful new species. A lot of work is being done currently also on the anatomy. We are focusing first in species that have some interest from the point of view of folk medicines or even poisonous or noxious weeds to help differentiate these species, but also we are moving towards like this work here on the left that was published right today. And here, Walter, that is a PhD student of Jellyman from, he did a wonderful anatomical work where he investigated all the vegetative traits from leaves and stems of all the species of section of subgenus Corydifoli. So this is the first subgeneric wide coverage level study comparing the anatomy of a subgenus of Baccharis, and he could find several traits that are unique for some species or some combinations of traits that help to differentiate some groups of species. And I also have a collaboration with a researcher from Rio de Janeiro Botanical Garden, Claudia. She is advising a PhD student who is currently doing electronic microscopy to understand how the tooth-tip indumentum evolved and how is the ontogeny, the development within the different subgenera of Baccharis. And maybe a lot of you know, several species of the genus, they have economic importance because they are used to as beverage, as infusions to treat some diseases. Some species are the source of the green propolis. That's a kind of byproduct of the honey production by bees. And the bees, they harvest some trichomes and the style apex of the Baccharis dracunculifolia female florets. And these structures have a high concentration of useful chemical compounds that make this product a promising compound that's being evaluated for treating several diseases, including cancer. We also have the species in the subgenus Corridifolia that they have associations with some fungi, and based on this association, they produce macrocyclic glycoticenes that are really toxic to cattle. And it's very common in southern Brazil, Argentina, and Uruguay to have problems caused by intoxication of cattle when they feed on these species known locally as mil. So here it's a metabolomic diagram where Rosana Casotti from Universidade de São Paulo, she did a metabolomic profile of 300 species of Baccharis. And it's very interesting that several groups retrieved in the phylogeny based on molecular data, they were confirmed by some characteristic compounds. Even that section aphyli, that's very variable concerning morphology and habitats occupied. That section have some shared compounds that distinguish it from another sections, for example. I'm also collaborating with people from phytochemistry to identify specific compounds and even new compounds found in some Brazilian species of Baccharis. And all this type of collaboration with people from mostly pharmacy and medicine could give origin to new drugs, for example. Here are some examples of Baccharis trimera being used to improve gastric ulcer healing, or the use of Baccharis dracunculifolia essential oils as anti-inflammatory compounds. So there are several studies made with several species of Baccharis looking for applications, from sunscreens to drugs to treat diseases just like HIV or even COVID. Collaborations are being done too in the area that I am more interested in directly in, that's the taxonomy. So here it's an example of the taxonomic review of Baccharis section Colopteri. This collaborative work with Mauricio and Victoria started with the master thesis of Victoria, where she did the flora of this section to Uruguay. And now she is expanding her work to review the whole section, and with the collaboration with Jennifer to use NGS to understand the relationships within species in this section, and also in the section aphyli, that's that was the one that in the past was considered together as the same section of Baccharis Colopteri. So that's it I have to say today. Thank you very much for everyone here interested in listening a bit about Baccharis. This work would never be reality if I did not have the support of all these agencies listed alphabetically here, and without the help of a lot of people that is here today and have collaborated from field work to lab and analysis and elsewhere. So thank you very much for your attention.
Thank you, Gustavo. I think everybody's clapping at their home, at your houses. That was an amazing talk, and we already have one question on the chat. If Ed Schilling, if you want to ask your question out loud, or someone can read it up. Can you see the question, Gustavo?
So just curious about subcellaribs, and you see those in different groups of Asteraceae, but I've never seen anything about what their adaptive significance might be. Is this just a packing thing in the head, or is there more to it?
Sorry, could Carol help me with the translation?
Yes. Pergunta sobre as costelas das sepselas, se tem alguma significância adaptativa ou se reflete alguma outra propriedade do desenvolvimento das plantas.
Well, I haven't seen any article so far dealing with the ribs number in the sepsela of Baccharis, but besides the ribs number, there is variation mostly in the mentum. So depending on the group, there are sepsela that are glabrous, and there are others that have other types of trichomes. And I think this, in conjunction together with the ribs, could have an ecological significance. By the way, I and Mauricio, we were talking about also the pappus, because the pappus differs a lot depending on the group we are focusing on. There are pappus that are accrescent, there are others that they are deciduous, the number of series varies. So by now, I don't know any reason for the number of ribs. I always thought some relationship with the vascularization, but I don't know if there is another side effect of this trait besides the vascular tissue.
Yeah, that's a great question. I never thought about what the number of ribs might mean and why they vary. Does anybody have any other questions for Gustavo? Go ahead, Mauricio.
I want to make a comment, just following up on what Gustavo replied to add on that question. I think it's very important to look closely at not just one structure, but also, the correlation with other structures to, find possible explanations on what the, evolutionary or ecological significance of those are. And just to show how this whole pandemic has, helped us to, make our community to grow. Gustavo was mentioning our work with the pappus, and there are two people there, Pecha Chankovic and Milan, who are from Serbia, who are going to be, collaborating in a whole team that we put together to, better understand the pappus and show some light on, the evolutionary significance of that. So I guess that once we start to have a more clear idea on how the structures are more precisely defined and they are plotted across the phylogeny, I think we will be, well, better placed to start answering those questions. But I think that those are, really, very interesting questions, because they will help us to understand how this, wonderful family evolved. So it's cool.
Even when some anatomical work that has been done on the sepsela of subgenus Baccharis, the results show that there are two very distinct groups within that subgenus concerning the types of cells that made the sepsela wall, for example. So I think there is a lot of interesting questions hidden within the fruits of Baccharis and Asteraceae as well.
Gustavo, I had a question. I was wondering if you had any ideas about why in certain places Baccharis will have micro-endemic taxa that are narrowly distributed. Here in California, we have a few, but they don't seem to be, like other types of composites, restricted to specific types of rock substrate or something like that. They just seem to show up in specific areas and not in others. So I'm curious, based on your comprehensive knowledge of the whole group, whether you have any ideas there.
Thanks for the question. The patterns I have seen is that these narrow endemics, most of them are linked to mountain summits. or in the case of California, some of them to certain chains, mountain chains that are not so high, but they have microclimates, for example. And there is also some of them, like one that is found in the coast of Rio, that's linked to, it's an island of different climate where there is a cold stream coming from the deep ocean that makes that part of the tropical coast of Rio looks like a scrub with very thorny vegetation. So I think it's mostly linked to the climate conditions. And then maybe these neuroendemics today, they were wider spread in the past, but as climate change with age, some of them get restricted to only specific niches where they have been evolving.
Any more questions? Okay, so if there's no more questions, we can start Dr. Itziar talk, and Isaac is going to introduce her. So
for the next talk, we have Professor Itziar Arnelas Seco from the Universidad Técnica Particular de Loja in Ecuador. This is an extremely interesting and biologically diverse area where there's a lot of diversity of Baccharis, which is what she's going to talk about. Species diversity of the genus Baccharis in southern Ecuador. So with that, take it away, Itziar.
Hello, thank you, Isaac. Thank you. And thank you to Carolina, and Mauricio, and Jennifer for helping us to organizing this nice place, and Gustavo for the invitation. So thank you for this moment. So sorry, a little, just a minute, please. So, I am going to speak about the species diversity on the genus Baccharis. So as Isaac told, I am Itziar Arnelas. So I work in UTP, in Universidad Técnica Particular de Loja, in the south of Ecuador, so next to Peru. So I am going to speak a little bit about the species in South Ecuador, because just I have studied in Loja and Zamora Chinchipe especially. So south of Ecuador is a really interesting place because it belongs with North Peru to Huancabamba, Amotape Huancabamba zone. So this place is really rich in his biodiversity, and especially the south of Ecuador has the 66% of species of Baccharis. So this is really a high number. So it's really diverse. So it is quite interesting because maybe it could be new species or many species that has been cited for this region maybe are not so clear. So as Gustavo said, Baccharis is characterized for tufted indumentum and dioici, and are really important from medicinal, people use really for medicinal uses. So they are really important for people here in south of Ecuador also. For example, Baccharis latifolia is really a species that grows here, and people use for stomach pain and things like that. So south of Ecuador, especially Loja and Zamora Chinchipe, has a different type of ecosystems, and in all type of ecosystem we can find Baccharis, for example, in Montanello forest in Zamora Chinchipe, with really with important influence of the Amazonian. We can find them, of course, in the montane forest at higher altitudes, and of course they are also usual in shrub paramo, but even we can find in dry shrub in Loja. Not many species. For example, we can find in Catamayo or different parts where the dry shrub are present. We can find there Baccharis nidifolia, for example. No, there are not many species, but Baccharis nidifolia is quite common in those dry places. So I used the phylogeny of Gustavo, sorry, but I think it's really a nice tree in order to explain how many species I have collected in Loja and Zamora Chinchipe. So subgenus Oblongifolia is just only represented for one species, and subgenus Molina is represented by more species like Baccharis auriculigera, Baccharis decusata, Baccharis latifolia, Baccharis salicifolia, Baccharis pedunculata, Baccharis quitensi, Trinervis, Baccharis wairacahensis, Baccharis volubilis, and Baccharis nidifolia that are now in different sections. So for south of Ecuador are 23 species, and those are the species that I could collect in Ecuador from those subgenus. The species that are in box, I couldn't collect yet. And subgenus Baccharis is better represented in Loja and Zamora Chinchipes in the south of Ecuador. So we have Baccharis reticulata, Baccharis sagittalis, Baccharis balnearia, Joensis, Baccharis sinuata, Baccharis buxifolia, Baccharis alpina, Baccharis genisteloides, Baccharis chilco, and Baccharis nitida, and Baccharis budlejoides, and also Baccharis tenuicapitula. From subgenus Baccharis, I couldn't collect Baccharis tenuicapitula, and even Baccharis budlejoides is quite a difficult species to me, at least, and I need just help to identify it. So I studied in Loja and Zamora Chinchipe because in the coast of Ecuador, in the south, in the province El Oro, we haven't got so many Baccharis. Indeed, in the Loja Herbarium, at CUTPIL, even QCE, I couldn't find so many species in El Oro, in the El Oro province, that is here in the coast of Ecuador. So the most species are in the Andes, in Loja especially, and some of them are in Zamora. So also I collected 4 species more that I don't really know the name yet, so I hope that we can study these 4 species. So to identify a species in Ecuador, and also of course in South Ecuador is quite difficult because we have not any complete taxonomic revision for the genus in Ecuador, as Gustavo said. So this is quite complicated. So I am going to speak just a little bit about some morphological characters that can be helpful for identifying some species. And I am going to speak about them, and I'll give some examples. So, for example, the avid could be a really interesting character because, for example, we have procumbent rabbit like Baccharis alpina. We have so many rabbit like Baccharis buxifolia. This is the most usual avid in South Ecuador for the species that are here, but also we have Baccharis volubilis, scanded rabbit, and Baccharis sagittalis that is an herb, sometimes could be at base. But also we have some trees like Baccharis balnearia, but this is not so common. And I am going to speak also about leaves morphology. Leaves morphology can be difficult, for example, in Secta andina. Baccharis joensis is a representative of Secta andina, but for in other sections could be useful to identify a species. For example, Baccharis alpina has really small leaves, spatulate or obovate. So Baccharis joensis has bigger leaves, of course, with pinnati nerves. And Baccharis decusata subspecies Kelski, that this is a member of subgenus Molina, has 3 nerves leaves, and Baccharis sagittalis, for example, has small leaves, at least the population that have collected with winged stems. So the shape of inflorescence, of course, is also maybe quite difficult for some species, but here in south of Ecuador, it's useful because, for example, Baccharis balnearia has Baccharis balnearias, for example, is the tree that I have saw. This tree is always I have collected in National Park of Yacuri in the south of Ecuador next to Peru. So it's really a beautiful tree, and the inflorescence are paniculate. But for example, in Baccharis pedunculata from subgenus Molina that I have collected in Zamora Tintipe at low altitudes in montane forest with really important Amazonian influence. The inflorescence has really few capitulae compared with Baccharis balnearia. Baccharis reticulata, for example, has sessile capitula borne in spikes, lateral spikes, and this species that I have number as 1 because I have not the name yet, for example, has capitula nearly solitary or born in maybe 2, 3, no more than 3 in a single peduncle. So Baccharis decusata, for example, has a really panicula inflorescence pyramidal and paniculate that is quite easy to see. So the shape of the volucre is not really important, at least for the species that are present here. Always is cylindrical or campanulate or cup shape. But for example, for this species that I have numbered as 1, the width of the capitula is really this is really big, you know. So if we compare with the Baccharis sinuata, that is a species that is present in south of Ecuador mainly, and I think in North Peru also. I saw this species because at least morphologically and the preliminary tree that I have constructed with molecular data, so that this species is the most closely related. So capitula here are born in the terminal branches, and they have a really short peduncles in the panicle. So I think it's so interesting these 2 species, no? We need to see. And as Gustavo and Mauricio was talking, the pappus is really interesting also. For example, Baccharis pedunculata has a really long pappus, more than the female flower. This here in populations from south of Ecuador, and even in Baccharis nitida, I saw that the pappus is deciduous, so it's also really interesting characters. The style branches from staminate flowers are also useful to identify a species. For example, Baccharis sinuata has capitate style branches, and Baccharis oblongifolium has similar capitate shape, but at least here in the specimens we can see this separation of the branches. And Baccharis quitensis, for example, from subgenus Molina, is an example of lanceolate style branches. So I studied also type of trichomes, and I made some pictures in the electron microscopy. For example, as Gustavo told us, some members of Baccharis has not tufted indumentum, like Baccharis decusata subspecies Helski, that has, I didn't see tufted indument. This species has pedestal trichomes, but most of the species in Ecuador, I mean, all species except the Baccharis decusata has tufted indumentum. For example, in Baccharis reticulata, we can see glandular biseriate trichomes with uniseriate flagellate trichomes. But for example, in Baccharis alpina, I could see that the arrangement of the biseriate glandular trichomes and uniseriate is different. So there are so many glandular biseriate trichomes with uniseriate in the middle, with I don't really know here because they are a little bit broken. So maybe glandular flagellate trichomes. So I need or we need to study this variation between species more deeply. So I saw two species from subgenus Molina related with the stones. In Baccharis nidifolia, I could see that epidermal is like striated with word cell quite smooth, but in Baccharis gitensis, the epidermal surface is not as the Baccharis nidifolia, and even the word cells are not as smooth as I can see in Baccharis nidifolia. So related with molecular studies, so I have 47 populations that belongs to 20 species, 20 different species, and with 4 species that I don't really know the taxonomic circumscription yet. So most species are amplified for this region that Gustavo works in his phylogeny. So 13 species I could amplify for 3 regions, and just only 2 I have for the 4 regions because ITs is quite ETAs is quite difficult, but I think that I have solved the problem. But I constructed preliminary eco tree and many relations that Gustavo recovered. So some of them are here also in this tree. For example, Baccharis reticulata is, sorry, I don't know if the secta Pille. Baccharis reticulata is within the species from this section, and for example, in this place, there are so many species of those sections from subgenus Baccharis, and I would like to show you this because also, of course, we need the region, the 4 region in order to corroborate those relationships. But those 2 collections that I don't really know the names, of course, and the names Baccharis angelica and tricuneata are not, of course, but appeared related with Baccharis odorata. Of course, this statistical support that is bootstrap is not so high, but if we see, for example, the picture of this collection, that is 3, 4, 5, 6 is here, and this is a shrub that is growing in the Paramos in south of Ecuador, and this is the type material or Baccharis odorata from Colombia, so it really looks like. And at first I thought that maybe it was something similar to Baccharis angelica, but this is not really Baccharis angelica, of course, but I think that Baccharis odorata could be related with this collection because I think it's morphological similar. So this is quite interesting because no species of Sectes pitose was previously cited for Ecuador. So I think that molecular study is really important to understand taxonomic circumscription of the species. So some conclusions to end this small talk, that for example, some species from secta andina like Baccharis joensis or Baccharis sendalensis are really complicated, at least to me, because morphological variation in the shape of leaves is important, and we need to study deeply. And morphology is so helpful, of course, in order to understand the phylogenetic relationships and micro characters. I think is so useful to species segregation, and I think it's really important to study, of course, all species as a whole, but this is quite difficult. But for South Ecuador, North Peru, I think is so important because both region belongs to Huancabamba and Motapezon that has so many endemisms in another species, and I think it's really important, of course, international collaborations, and in this sense, I really thank you very much, Gustavo, for your help in the molecular study and molecular morphological study, and of course my university, because I had 2 small projects in order to study morphologically and molecular this species in the South Ecuador, and of course the cited herbarium. Thank you all of you.
Thank you, Itziar. That was an amazing talk, and it's so impressive that a small country like Ecuador has such an amazing diversity. I imagine there's still a lot of work to be done for the Ecuadorian botanists.
Yes, sure. See, of course, and the taxonomy is not well resolved in so many groups, not only.
Yes.
Yeah. So,
Does anybody have any questions for Itziar?
I have a question.
Yeah.
So it's how many species left to get all the Ecuadorian Baccharis sampled?
sorry, Gustavo, again, please.
Gustavo, you can speak in Spanish if you want.
¿Cuántas especies faltan para tener un muestreo completo del género en Ecuador?
Pues como he colectado unas 10, pues unas 20, creo que como unas 18 más, porque en principio hay 38. Entonces, ajá. I think
So,
more, maybe 40% of the species I need to collect. Mm-hmm.
And what's the main constraints for getting these samples in Ecuador? Bureaucracy or getting in the place where the plants grow?
Both. I
Both.
think bureaucracy maybe is the most important issue, because we need so many permission from government in order to extract DNA. So, but I think now it's easier, but we will see. Things change in related with bureaucracy, and maybe it's easier, but right now it's not easy. And financial support also is quite difficult sometimes, you know. And now there are so many problems.
we have a question in the chat. Milan is asking if there is a published paper about the development of the glands that you showed.
So that glands, they were first described more than 1 century ago in a optical microscopy paper. And since then, I haven't seen so many efforts trying to understand how they evolved. There is a guy doing his PhD with electronic microscopy scan in Rio de Janeiro Botanical Garden. He is sampling the 7 subgenera, 1 species at least per subgenera, in different developmental stages. And he is looking for, based on the development ontogeny and the placement of every species in the phylogeny, to figure out how this special type of glandular trichome appeared. It's funny because when you look at some plants of Baccharis in the naked eye, they sometimes look like myrtaceae that have that punctate glands. Some of them are really, really conspicuous. You can see huge glands with the naked eye.
So, Gustavo, you saw that phytochemistry was important characters in order to understand I don't know how to say in English, but it was phylogenetic signal.
For a section acid, for example, we have that number of cipsellar ribs as a morphological character, but there are not many other traits useful to recognize that group. And when you look at the plants, some species grow in wetlands, others are from cerrado, that's a savanna, or very edges of forest. So the habitats are very variable. And when Rosana Casotti from Universidade de São Paulo did a wide sampling for metabolomic study, she found some compounds that were only occurring in that section. But now for she to identify these compounds, it's a bit more complicated. She found the signal, she can infer what's the likely compound, but then she needs to go further and get much more sampling so she could identify exactly which compound is that. But not only for that section, but for other sections too. There are some chemical compounds that are useful to circumscribe some sections that even don't have any morphological character, for example. So far.
Okay, because I saw that, for example, as I explained that in Baccharis reticulata, the glandular biseriate trichomes are not many, but in Baccharis alpina, there are a lot of those type of trichomes. So it's quite interesting.
So Mauricio just reminded us that Capitulum is accepting articles for publication until October 15th, and to get in touch with him if you have any ideas for articles. And I just posted a YouTube channel link on the chat. And also you can find all this information at composite.org. I want to thank Itziar and Gustavo again for coming to give this talk. They were very good. I hope everybody enjoyed it as much as I did. And if we don't have any questions, I think we can probably finish the session, and I'll see you all again in October. Thank you so much.
Thank you, everyone.
Thank you.